Poliomyelitis is a viral infectious disease caused by poliovirus. The name itself refers to inflammation of the gray matter of the spinal cord and primarily affects motor neurons. Polio became one of the most feared diseases of the 20th century because of its association with sudden paralysis, particularly in children. Now, what makes polio unique is that severe disease is actually rare. Most infections are mild or completely asymptomatic. However, when complications do occur, they can be neurologically devastating. To understand why this virus behaves the way it does, we first need to look at its structure and classification. Poliovirus belongs to the family Picornaviridae and the genus Enterovirus. It is a non-enveloped, positive-sense, single-stranded RNA virus, and both of those characteristics are important for understanding how it behaves. When we say the virus is non-enveloped, that means it does not have a lipid membrane surrounding it. Instead, it is made up only of a protein capsid that protects its genetic material. This actually makes the virus more stable in the environment. Without a fragile lipid membrane, it can survive exposure to heat, drying, and even stomach acid. The stability is one of the reasons polio spreads so efficiently through contaminated water and poor sanitation. Positive sense means that once the virus enters a host cell, its RNA can function directly as messenger RNA. The host's ribosomes can immediately begin translating viral proteins without the virus needing to convert its genome first. This allows for rapid viral replication inside intestinal cells. So, the fact that polio is both non-enveloped and positive sense helps explain why it spreads easily and replicates efficiently once inside the body. Now, there are three serotypes of poliovirus, types 1, 2, and 3. All three cause the same disease and follow the same pathogenesis. However, they differ in their capsid protein structure, which means the immune system recognizes each type differently. Immunity to one serotype does not guarantee protection against the others. Historically, type 1 caused the majority of paralytic cases and is the only remaining wild serotype still circulating today. Types 2 and 3 have been declared eradicated in their wild forms. Poliovirus is primarily transmitted through the fecal-oral route. The virus is shed in stool and can contaminate water, food, or surfaces. In regions with inadequate sanitation, outbreaks may become waterborne when sewage contaminates drinking water supplies. In epidemiologic terms, contaminated water and food act as vehicles, meaning they serve as inanimate mediums that carry the pathogen from one host to another. Contaminated surfaces can also contribute to transmission. These are referred to as fomites, which includes objects or materials that can harbor infectious particles such as shared utensils or improperly sanitized surfaces. But most importantly, poliovirus is not vector-borne. Humans are the only natural reservoir, and there is no insect or animal vector involved in its transmission. It can also spread through close person-to-person contact and is less commonly through respiratory droplets. After entering the body, the virus replicates first in the oropharynx, which is the middle part of the throat, and the intestinal tract. In most individuals, the immune system produces neutralizing antibodies that prevent further spread. However, in a small percentage of cases, the infection progresses beyond this initial stage. Polio has a wide clinical spectrum. Approximately 90 to 95% of infections are asymptomatic. Some individuals experience mild symptoms such as fever, fatigue, sore throat, or headache. A smaller percentage develop non-paralytic meningitis, which involves inflammation of the membrane surrounding the brain and spinal cord. But in about 1% of infections, the virus progresses to its most severe form, paralytic polio. Because polio is caused by a virus, there is no specific antiviral cure. Treatment is primarily supportive and depends on the severity of disease. For mild cases, management includes rest, hydration, and symptom control. In cases of non-paralytic meningitis, supportive care and monitoring are typically sufficient. Regarding paralytic polio, the destruction of lower motor neurons leads to flaccid paralysis, meaning the muscles become weak and limp rather than rigid. Reflexes decrease, and over time, muscle atrophy develops due to denervation. In some patients, the virus affects the motor neurons that control the diaphragm and intercostal muscles, which are the muscles responsible for breathing. When this happens, the patient develops respiratory paralysis. And again, because there is no antiviral cure for polio, treatment in these cases are also supportive. One of the most significant medical interventions used during the height of polio epidemics was the iron lung. The iron lung was a negative pressure ventilator, which will be discussed more in our history slides. One important factor in the clinical spectrum is age. Polio most commonly infected young children, which is why it was often called infantile paralysis. On this slide, I'm showing two graphs to illustrate age distribution patterns in poliovirus infections. The first graph breaks down cases by viral strain. Here you can see that approximately 86% of wild poliovirus type 1 cases occurred in children aged 0 to 3 years. Similarly, about 76% of circulating vaccine derived poliovirus type 2 cases also occurred within the same age group. These figures come from a global surveillance data published in eClinical Medicine, which analyzed age distribution patterns across multiple endemic regions. This strain-specific breakdown shows that regardless of whether the virus is wild type or vaccine-derived, early childhood remains the most effective age group. The second group simplifies the data by combining strain information and emphasizing the overall trend. In this version, you see that more than 90% of all reported cases in certain endemic settings occurred in children under 5 years old, with a median age of infection around 2 years. These statistics come from a peer-reviewed epidemiological study published in Tropical Medicine and International Health. Together, both graphs reinforce the same conclusion. Poliovirus transmission and clinical disease are heavily concentrated in very young children.
So, for the early history of polio, it was first identified in the late 1800s. Specifically, in 1894 was the first documented polio outbreak, and this left us with 18 dead and 122 paralyzed in Rutland County, Vermont. Then, a few years later in 1908, the cause of polio was identified, and they discovered it as a research from virus on monkeys done specifically. Then, in 1931, was the invention of the electron microscope, and 4 years later, in 1935, there were early trials on polio. These were very much unsuccessful and left many dead, paralyzed, or people with severe allergic reactions. Moving on the history of polio, we move on to the 20th century epidemic. In the 20th century, there was a peak in the 1950s, specifically 1952, where nearly 60,000 cases of polio were reported in the U.S. This led to the closure of many pools and schools due to the disease outbreak. In 1955, Salk's vaccines were released, and it was reported to be 80 to 90% effective against paralytic polio. However, there was a Cutter incident where the vaccine program was suspended weeks after the vaccine was licensed, and there were reports of vaccinated paralytic cases, and they also rose as well. In 1961, oral vaccine was licensed by Dr. Albert Sabin, and it was a live oral polio vaccine licensed to protect against type 1 polio. In 1963, there were oral vaccines that were protected against type 1, 2, and 3 polio. And in 1994, nearly 31 years after the original vaccines were created, polio was declared eradicated from the US. From roughly 1928 to the 1960s was known as the polio iron lung era, and the iron lung was developed by Philip Drinker and Louis Shaw in 1927. The mechanism of the iron lung was a sealed iron tank that had negative pressure created by a motor-driven diaphragm, and it essentially forced air into patients' lungs. For patients that had to deal with the iron lung, it was about weeks to months to years inside, depending on how long one lived. And there was constant care for your basic needs and need for physiological resilience and mirrors to see around. Essentially, these patients were trapped in the iron lungs almost 24 hours for 7 days a week, 365 days a year, and tended to live slight miserable lives. In the 1950s and 1960s, vaccines were created and they led to decreased use in the iron lungs, especially with less people getting sick, and they were replaced by modern positive pressure ventilators. The Polio Global Eradication Initiative began in 1988 and was led by the World Health Organization, the CDC, UNICEF, and Rotary International, and their goal was to complete global eradication in polio. The strategies that they used to eradicate polio included mass vaccination campaigns, the oral polio vaccine used for herd immunity, environmental surveillance such as testing sewage, and rapid outbreak response teams. And they were decently pretty successful from 1988 having 350,000 cases annually to today having over 99% reduction in cases. This means that wild polio virus is eliminated in most countries now.
There are two main vaccines used for polio throughout the world. The first being OPV. OPV, which is oral polio vaccine, is no longer used in the United States due to it being deemed risky, as it causes VAPP in one case out of 2.4 million doses. VAPP is vaccine associated paralytic poliomyelitis, which is a rare severe side effect of the OPV that causes paralysis in its recipients. The second vaccine is IPV, or inactivated polio vaccine. IPV is a shot given in the arm or leg, depending on the patient's age. Children should receive 4 doses total, the first at 2 months old, the second at 4 months old, the third at 6 to 18 months old, and the fourth at 4 to 5 years old. IPV is the only polio vaccine in the United States and has been used since the year 2000. OPV is a mixture of 3 strains of polio. The strains are saving strains type 1, 2, and 3. Polio is subject to genetic variation, causing a neurovirulent strain. It is responsible for VAPP and VDPV. VDPV is vaccine-derived poliovirus. 2 categories of this is immunodeficiency associated and ambiguous. VDPV becomes a higher risk once wild poliovirus disappears. Wild poliovirus is a naturally occurring non-modified strain of the virus that primarily affects unvaccinated children and is highly infectious. Under-immunized areas with population clustering are more susceptible to CVDPV. Due to this, some countries have switched to an IPV rather than OPV, which protects against paralysis but is not as effective at stopping gut replication and can still have a lot of transmission. In addition to this, acute flaccid paralysis, which is also caused by polio, to environmental surveillance is critical. This image shows the type of vaccine that IPV and OPV are, the immunity they both provide, the status of their herd immunity, and the risks that come with them. IPV is an inactivated vaccine, which means that it cannot replicate the virus and cause disease, making it non-infectious. OPV is a live vaccine, meaning it has a live weakened version of the virus, causing a natural infection, but provides a long-lasting immune response. IPV and OPV both provide immunoglobulin G immunity, which is systemic immunity, and OPV also provides immunoglobulin A immunity, which is mucosal immunity in the stomach. The herd immunity of IPV is limited, meaning a significant portion of the population is immune, which reduces spread but is not entirely stopped. High-risk groups can still become infected. OPV, on the other hand, has a strong herd immunity, which means a high percentage of the population is immune due to prior infection or vaccination, which reduces overall risk of infection for everyone, including those not immune. IPV has no risk that comes with it. OPV, however, has a rare reversion risk, meaning that the disease is supposed to prevent, it can also cause it.
Salk created the inactivated polio vaccine in the early 1950s, and it was licensed for medical use in 1955. 2 years after the introduction of the vaccine, the yearly average amount of cases had decreased by 2,000 cases. This solution led to an exponential decrease in polio cases that caused the United States to be considered eradicated of polio by 1979. Poliovirus has had 3 strands, wild types 1, 2, and 3. In 1999, wild type 2 was eradicated, followed by wild type 3 in 2020. The vaccine was able to help stop the spread of polio because it decreased the risk of both infection and transmissibility. Had the vaccine only been capable of stopping either the infection or the transmissibility, it wouldn't have been an effective method to begin working towards herd immunity. Though poliovirus is eradicated in the United States, it cannot be truly eradicated until it is no longer present in any country. At this time, wild poliovirus type 1 is still seen in Afghanistan and Pakistan. International trade and travel with these countries leaves the rest of the world at risk for polio's reemergence. The herd immunity threshold for poliovirus has a seemingly wide range. For wealthier communities, the herd immunity threshold is about 75%. This means that 75% of the community would need to be immune, most likely through vaccination, to stop the spread of poliovirus. In poor populations, the herd immunity threshold must be closer to 97% immunity to prevent the spread of the disease. The threshold differences are dependent on the differences in the environment. In wealthier communities, there is more likely to be an emphasis on hygiene and sanitation than a community that doesn't necessarily have the funding to spend on disinfection and cleanliness. This graph from the National Library of Medicine shows that the United States was able to reach 80% immunity in 1955. The graph also shows us that Burma had a herd immunity threshold of about 97% and did not reach that threshold until 1979. By the time Burma met the herd immunity threshold, polio was eradicated in the United States. We can see here that in Burma, there was an average of 30 people infected by one infected person before the introduction of the vaccine. That's the R0 value. Because their population was fully susceptible to infection, the herd immunity threshold was much higher. Burma's placement on this chart is evidence of how third world countries, who cannot focus specifically on disease prevention, will have a higher R0 value to combat and therefore have to reach a higher herd immunity threshold.
Wild poliovirus, or WPV1, is endemic in Afghanistan and Pakistan. Humans in these areas are the only ones that contract this disease, and it just continuously reoccurs in the local population. Some of the reasons for this is that these countries in the Middle East, a lot of times they don't have the proper vaccine coverage that they need, so they're not able to reach that herd immunity threshold, and they're usually in war zones or places to where people can't get the proper health care that they need. And also, they have poor sanitation, whether in culture or just financially, whether they're able to provide for that or their government's able to provide for them. Poliovirus reached its peak around the 1980s, in the early part of the 1980s, and the vaccination was introduced heavily around 1988. And based on this graph, you can see that any trend after that, it just decreases. And especially after that 1988, there's a significant drop in the number of cases worldwide. Right now, there's very, very little cases in poliovirus due to all of these different countries over time, with the trend in the graph reaching herd immunity due to the vaccination efforts that each country has set in place. In July 21st of 2022, the United States received its first poliovirus diseased person since 3 decades, which this goes to show how well the vaccine did work, as this person was traveling from outside the United States to the United States. He was Hungarian and he resided in Rockland County. This case was confirmed by the New York Department of Health, and they had received other persons that also had poliovirus, which again were traveling from outside the United States to the United States. This virus has been detected to have a vaccine-derived poliovirus, which is type 2. This suggests that the origin from the live plus the OPV vaccine just wasn't quite getting the poliovirus eradicated, and is more of so a hybrid. Again, immunization with the shot is the best case to prevent poliovirus from continuing.
Even though polio cases have decreased dramatically worldwide, that progress is only possible because of strong surveillance systems. There are two main types of polio surveillance. The first is acute flaccid paralysis, or AFP surveillance. Since polio can cause sudden paralysis, health officials monitor any case of unexplained paralysis in children under 15. When a case is detected, stool samples are collected and tested in a lab to confirm whether poliovirus is present. There are two key performance indicators used to measure how effective AFP surveillance is. The first one is non-polio AFP rate. This helps to ensure the system is sensitive enough to detect possible cases. And the second is stool adequacy, which is 2 stool samples are collected within 14 days of symptom onset and are properly transported to a lab. If these standards aren't met, cases could be dismissed. The second major system is environmental surveillance, which involves testing sewage samples for poliovirus. This is extremely important because polio can circulate silently, meaning that it can spread in people without showing any paralysis symptoms. Environmental surveillance also measures something called the enterovirus isolation rate, and this ensures that sites are sensitive enough to detect the presence of the virus. Together, AFP and environmental surveillance allow for early detection, faster containment, and have significantly reduced overall poliovirus transmission. There are 3 wild poliovirus types: WPV1, WPV2, and WPV3. Thanks to global vaccination efforts, WPV2 was declared eradicated in 2015 and WPV3 in 2019. Today, only WPV1 remains, and that type is endemic in Pakistan and Afghanistan. However, as wild polio decreases, vaccine-derived poliovirus, or VDPV, becomes more significant. The oral polio vaccine contains a weakened live virus, and in some instances, this weakened virus can circulate in under-immunized populations and mutate over time. When it regains the ability to cause paralysis, it becomes circulating vaccine-derived poliovirus, or CVDPV. VDPV is genetically distinct and can cause paralytic disease just like the wild polio virus. For example, in Nepal in 2024, a vaccine-derived strain was detected. This shows that even as we approach eradication, surveillance and high vaccination coverage still remain essential. Polio is a really good example of the One Health framework. Poliovirus only infects humans, meaning there is no animal reservoir. This makes the virus easier to eradicate. However, many of the individuals that are infected are asymptomatic, so these individuals can unknowingly transmit the virus, contributing to silent transmission within communities. From the environmental perspective, the virus spreads through the fecal-oral route. So in areas with poor sanitation, open defecation, or inadequate wastewater treatment, contaminated water becomes a transmission pathway. While the virus does not survive forever, it can survive long enough to infect new hosts, especially in densely populated areas. This is why wastewater surveillance has become such a powerful public health tool. Detecting poliovirus in sewage allows officials to identify circulation before paralytic cases even appear. Infrastructure also plays a key role. Countries with strong sanitation systems and high vaccination coverage are much less likely to experience sustained outbreaks, even if the virus is introduced to the population. In contrast, regions with poor infrastructure and gaps in immunization coverage are more vulnerable. So the One Health connection here shows that polio eradication depends not just on vaccines, but also on sanitation systems, environmental monitoring, healthcare access, and coordinated global policy.